Hello everyone, welcome to MGI Webinar Plus series. In the last Webinar Plus video, we explained the operation of MGISP-100B based on the use of MGIEasy magnetic beads virus DNA-RNA extraction kit in details. So, in this video, we will continue with the previous theme to provide the operation training of MGISP-100 along with another specification extraction kit, MGIEasy magnetic beads virus DNA-RNA extraction kit with 96 reactions. MGISP-100 can extract 8 to 32 samples per 40 to 80 minutes. The flexible throughput makes it be more applicable to small and medium-sized research laboratories, such as hospitals. So today, let's focus on the second choice of MGI virus nucleic acid extraction solution, the combination of MGISP-100 and MGIEasy magnetic beads virus DNA-RNA extraction kit with 96 reactions. Before this Webinar Plus video, just remind everyone to comply with the requirements of the National Center for Disease Control and Prevention or local laws and regulations when performing virus extraction operations, and it's necessary to carry out the extraction of virus nucleic acid in the appropriate laboratory and wear personal protective equipment as required. So, the objectives of this Webinar Plus video are hoping that you can master the hardware structure of MGISP-100 and master the automated extraction process of basic throat swab samples. First of all, let's have a look at the specifications of MGISP-100. It's a medium throughput automated extraction system and a liquid handling workstation, which is much smaller than MGISP-960 in size. Its height is 777 mm and the length is 780 mm. The width is 725 mm and requires just less than 0.5 cubic meters total floor space. In addition to the automatic equipment, MGI will also accompany the machine with a computer, built-in MGISP-100 control software. Customers do not need extra computer. After the external specifications, let's take a look at the environmental requirements. The power of this automatic device is 1,600 watts. And the machine weighs 130 kilograms. For the laboratory environment, it's required to control the temperature between 19 to 25 degrees, and the humidity should be kept between 20 to 80%. The pressure should be between 80 to 106 kilopascal, and the highest elevation that can be supported is 2,000 meters, and the level 2 cleanliness is recommended. Next, let's go to the internal structure of MGISP-100. Despite its small size, MGISP-100 is still highly integrated with various functions. The automated cleaning components, UV lamp and air filter unit, and HEPA system of positive pressure. Filter efficiency is up to ISO Class 5. The 8-channel high-precision pipette and various positions with different functions, such as the temperature control module and the purification module, and the PCR module is also equipped inside. After the general introduction, we can keep going to the main part of this video, the workflow of MGIEasy magnetic beads virus DNA-RNA extraction kit in MGISP-100. We show here is the process of automated extraction on MGISP-100 with MGI extraction kit. First of all, we need to do the automated pre-clean. During the cleaning time, operators need to prepare samples, reagents, and consumables. Unlike MGISP-960, all preparation work of MGISP-100 can only be finished manually. After all preparation, the automatic extraction process can be started. Because MGISP-100 can support different sample numbers, so the extraction time varied from 40 minutes to 80 minutes. After the extraction, operators need to collect the extracted RNA products and place them in the -80-degree fridge or directly conduct the subsequent RT-PCR reaction. Finally, a post-clean of the instrument is required to ensure the cleanliness of the machine, so as to reduce the risk of cross-contamination between benches and the infection of operators. The kit we demonstrate in this video is MGIEasy magnetic beads virus DNA-RNA extraction kit, which is enough for 96 reactions and is for research use only. The corresponding automatic protocol is MGISP-100 MGIEasy magnetic beads virus DNA-RNA extraction kit instructions. The solution we used in this video is JB806026 MGISP-100 MGIEasy magnetic beads virus DNA-RNA extraction RV1.0 SV1.4. Before you run the script, make sure you are using the latest version of the solution. In this solution, 4 scripts of different throughput are provided. According to your actual sample number, select the corresponding script. And for the last script is used for prickling, which is specially for the prickling of COVID-19 extraction process. We increase the radiation time of UV lamp to decrease the cross-contamination between benches and reduce the risk of virus infection for medical staff. Next, we will explain the operation process and the precautions of each step in detail. Please ensure that you have carefully read relevant reagent kit user manual and the automated instructions before viewing the following course. The first part is the preparation before automated extraction. Firstly, preparing the device. Before first use, please install the application scripts on the MGISP-100 according to MGISP-100 and MGISP-960 application script installation instructions. Then the equipment should be cleaned in advance. Double-click MGISP-100, select Real Mode, and then click Create. Next, click User Entry. Enter the initialization interface, click Initialize. The initialization takes about 2 minutes. When the interface shows Initialize successfully, you can go to the next step. Click the Menu button and select Wizard. Choose Solution MGISP-100 MGIEasy magnetic beads virus DNA-RNA extraction, and select the MGISP-100 pre-clean script. Since the COVID-19 is highly infectious, we increase the exposure time of UV lamp to 30 minutes. Therefore, we need to run this specific pre-clean script instead of the normal pre-clean program. Then click Start. During 30 minutes cleaning time, we can prepare the consumables, samples, and the reagents. Secondly, we need to prepare consumables. This table shows the corresponding consumable needs for different throughput. According to your sample number, take out the consumables required for one workflow. And in this video, we will take 32 samples per run as example. So in the following, all operations and reagent volumes are based on 32 samples per run. For other throughput, please find the corresponding instruction in the automated menu. For 32 samples, it needs 2 boxes of 250 microliter automated filter tips, 1 plate of 1.3 ml U-bottom deep well plate, 8 strips of breakaway 96 wells clear tubes, and 8 strips of flat optical caps. Thirdly is sample preparation. Depending on the sample type and the requirement of local CDC, perform sample inactivation firstly. The automated sample preparation system can process 32 samples at one time. So, we need to take 4 strips of clear PCR tubes and transfer 32 samples in turn. Add 180 microliter inactivated samples to the prepared strip PCR tubes in the BIC. Ensure that no air bubbles exist at the bottom of the tube and no liquid remains on the tube wall. After transferring, close lids and place tubes on ice. Then, use a mark pen to label each strip. Final preparation work is about the extraction kit. The extraction kit we show here is MGIEasy magnetic base virus DNA-RNA extraction kit with 96 reactions, which consists of 2 boxes. Box 1 contains buffer MW1, buffer MW2, buffer MLB, anis-free water, magnetic base M, and proteinase K, which is generally transported at room temperature. After receiving the kit, please store proteinase K and magnetic beads at 2 to 8 degrees, and the remaining reagents can be stored at room temperature under dry conditions. Box 2 is the enhancer buffer, which is generally transported in ice packs or dry ice, and can be stored in -20-degree fridge after receiving. Please note that absolute ethanol needs to be added into buffer MW1 and MW2 as indicated on the label of the reagent bottles before the first use. Take out 2 bottles of buffer MW1 from box 1. Prepare the absolute ethanol and pipette. Add the corresponding volume of absolute ethanol to the MW1. Then close the lid and reverse the bottle to mix thoroughly. Next, take the label on the bottle. And the same process for MW2, adding ethanol and taking the bottle. When MW1 and MW2 are ready, we need to prepare lactate and bending buffer mix. Since lactate and bending buffer is composed of 5 different reagents, and the proteinase K in them is easily inactivated, the best operation is to prepare and separate LNB buffer according to the number of samples within 30 minutes before the start of the extraction process. This table shows the volume of each reagent required for 32 reaction mixtures. Before preparing, take out the reagents from the kit and thaw them under room temperature. Mix them thoroughly before use. Thaw enhancer buffer at room temperature thoroughly and mix it by reversing for several times. Centrifuge shortly and place on ice for further use. Take a new 50 ml Falcon tube, label with LNB buffer, adding 5.12 ml of buffer MLB, adding 6.4 ml of absolute ethanol. Vortex the magnetic base M thoroughly and then add 480 microliter. Next, adding 480 microliter of proteinase K. And finally, adding 32 microliter of enhancer buffer into the Falcon tube. Close the lid and reverse the tube for several times to mix. All extraction reagents are ready. We can move to the reagent separation part, which is different between MGISP-960 and MGISP-100. MGISP-960 can support automated reagent separation, but for MGISP-100, we need to separate all reagents by hand. Various extraction reagents are arranged in different columns. Take a new deep well plate and use different color markers to label the corresponding columns. Prepare some clean plastic liquid containers, the corresponding range single pipette, strip pipette, and tips. For hardness-free water, we recommend using single pipettes to transfer the reagent from the bottle directly so that the contamination can be avoided. And for MW1 and MW2, we can pour the reagent into the clean plastic liquid container and use the strip pipette to transfer the appropriate volume to the corresponding columns. Please notice that different liquid containers need to be used for each reagent to make sure the rest of the reagent is clean so as to be poured back into the bottle for further use. Finally, for the LNB buffer mix, shake the 50 ml Falcon tube thoroughly before pouring into the clean container. Transfer 360 microliter to the corresponding vials, and then discard the left reagent directly. LNB buffer cannot be recovered for next use. After separation, check there is no air bubbles existing at the bottom of each vial and no liquid remaining on the wall. Then the plate can be sealed first and used within 30 minutes as far as possible. Once all the preparation work is done, we can start the second part, automated extraction process. The wizard interface, directly click Solution and select MGISP-100 MGIEasy magnetic base virus DNA RNA extraction. Click Script and select MGISP-100 virus DNA RNA extraction 32 reactions. Operation deck layout for 32 reactions will be displayed, and follow the on-screen instructions to place 4 empty strip tubes and position 1-1, column 1, 2, 3, 4 for extracted RNA products. Next, place 2 strips of sample tubes at position 1-1, column 5 and 6, and the other 2 strips need to be placed at position 5-1, column 1 and 2. Then put 2 boxes of 250 microliter automated tips at position 2 and position 4, and the 96 deep well plate within separated extraction reagents need to be placed at position 6. Finally, place the waste container with the medical garbage bag on position 7. After arrangement, check again according to the on-screen layout. Then we could click Start to let the automatic virus extraction process run. MGISP-100 is equipped with a 8-channel high-precision pipette, which can load 8 tips at one time. So the throughput it supports is usually the multiple of 8, such as 8, 16, 24, and 32. For different throughput, the robot pipette will load the corresponding column of tips to transfer samples to the extraction reagents. A different operation between MGISP-960 and MGISP-100 is that MGISP-100 is not equipped with the gripper, so the magnetic rack has been designed in the lifting mode. During incubation, the magnetic rack locates at the bottom, and when purification is needed, the magnetic rack will rise and attract the magnetic beads in the deep well plate. The extraction process of 32 samples is expected to run for 18 minutes. After the running, take RNA product strip tubes at position 1-1 and close the lids. It can be directly used for the subsequent RT-PCR detection or stored in -80 fridge. Then we need to go back to collect the left consumables. Dispose of used strip tubes, tips, and deep well plates to the designated waste area. The next batch is carried out immediately. Use 75% ethanol and dust-free wipe to clean the surface of positions and do a pre-clean. If no more experiment is to be conducted on the day, perform a post-clean before powering off the device according to MGISP-100 and MGISP-960 cleaning instructions. Above is the MGISP-100 MGIEasy magnetic base virus DNA RNA extraction workflow. In the process of operation, it's necessary to pay attention to the following situations that may affect the nucleic acid extraction results. Firstly, the following conditions might affect the quality of extracted nucleic acids, and the problems should be solved before performing the detection. First, whether the reagent is stored according to recommended storage environments. Second, whether the carrier is placed correctly during the running of workflow. And other misoperations might lead to unqualified detection results, such as expired reagents are used, the pipette is inaccurate, the room temperature is too high, or the detection is not performed according to the extraction procedure of the instructions. Alright, that's all of MGISP-100 operation training by using MGIEasy magnetic beads various DNA RNA extraction kit 96 reactions. Hope this video is helpful for your experiment. For more related videos, please subscribe our official YouTube channel, MGI International, or our official website. In addition, if you have any technical problems, you are also welcome to send your questions to mgiinfo@genomics.cn. MGI Global FS teams are very happy to help you. Thank you.